Welcome you all to this cryo-EM course. Last class we already discussed about how we can do different kind of imaging, and that time we discussed about the different kind of thick stain, light stain, and the proper staining, and we also discuss about the antigen antibody complex formation and why SCC purification is important and how we can isolate different kinds of molecular weight using the SCC techniques. Not only that, we also discuss about that lipid protein aggregation. So, lipid protein aggregation is an important part that when we are dealing with a lipid and protein sample, and as well as there is a protein protein interaction, as I mentioned that there is a lot of flexible region or intrinsically disordered region. Now, if our protein is very happy in presence of particular salt concentration or glycerol concentration, and suddenly if we remove all this glycerol or the salt concentration, what will happen? There will be lot of area of this protein will be exposed, which is probably buried somewhere. Now, if it is exposed, and then they will try to, rest of the protein molecules will try to come closer, and then when there is a disordered region, and then they will start coming together and then clustering, and it will completely aggregate, and from the aggregate we cannot go back to the normal condition. The lipid, in presence of lipid, that problem is more, and last class I already talk about little bit about the membrane protein also. The membrane protein generally stay inside the membrane. So, if you look at the screen, so here if I just draw how these lipid bilayers look like, this is our lipid bilayer, right. So, this is a charge head group, and this is our charge head group of the lipid, right, and then there is a this tail area of this lipid, mostly very hydrophobic. So, here if we consider there is a membrane protein, how this membrane protein it will stay, that membrane protein there will be a helix, and then it will go like that, it will stay within that membrane. Now, in this membrane protein, if we want to take out this membrane protein, what is the best way? That we are discussing previously, that most of the time that membrane protein, if you just consider that there is any pump, any transporter, that GPCR or that efflux pump or bacterial secretion system, all are membrane protein. They will stay inside the membrane, and they will be participate in different kind of cell signaling pathway, or that efflux pump or secretion system, they will release different kind of material. So, membrane protein is surrounded by that lipid head group and then this chain. Now, why they are surrounded by this? Because most of this helical structure or the beta strand, there are lot of hydrophobic residues are there. So, if you just consider these tails are hydrophobic, and the membrane protein in this amino acid residue top, there will be some polar residue, because they are going to interact with the other protein or ligand or with water molecules. But inside this region, inside this particular part, there is a lot of hydrophobic amino acid will be there. This hydrophobic amino acid will be surrounded by this hydrophobic residue of this lipid chain, and that is the reason membrane protein will be always surrounded by the lipid membrane. Now, when you will do the cell lysis to extract that protein, whatever is embedded in the membrane, with the membrane it will go to the cell pellet. In this particular case, you need detergent. So, detergent, if you just consider what is the difference between the lipid bilayer and the detergent micelle. So, detergent micelle also it will be surrounded. If I just do like that, this will be the detergent micelle. It also has a polar head group and this kind of hydrophobic. So, this hydrophobic amino acids and this polar head group. This polar head group, inside there will be a water molecule or something will be inside. This is called the detergent micelle. Whenever we are talking about membrane protein, if we use this detergent to solubilize this lipid bilayer, there is a high chance if we just use a very high concentration detergent, most of the time this lipid bilayer, this will dissociate. This will remove from the membrane protein and the membrane protein will be surrounded here within that lipid micelle, a detergent micelle, or some, when lipid will form a micelle kind of thing, it will stay inside that. So, if my membrane protein is surrounded by the detergent micelle, this will be a isolated one single membrane protein and that I can image. So, here I am using a membrane protein which has helix, that I draw total 4 helical structure, that okay, there is a helical structure of this membrane protein and it will surrounded by the detergent micelle. Now, what is the concentration of the detergent micelle? That is depending on how much protein do you have, what is the solubility and lot of other factors, it will play an important role. But membrane protein, whenever we will try to purify, detergent will be a extra component. Now, suddenly if you just consider that detergent is not very good thing for protein or any biological macromolecules, you want to remove the detergent. When you will remove this detergent completely from here, what will happen? This protein will be free and there is a lot of hydrophobic region will be free. Then there will be lot of hydrophobic region, they will come closer to each other and then they will start clustering with each other. And then they will form a protein aggregation. If these are the individual protein molecules, if we just consider from top, if we look, this is one single protein. So, if this is my single protein, they will come and cluster with each other. And when they will form this kind of cluster, it is very difficult to break that cluster. So, this is called protein aggregation. And if there is a protein aggregation, that will be completely useless. Now, if there is a helix, sometime beta strand can also come and form. So, this kind of beta strand you can see they are coming together and lot of time they form a barrel kind of shape. So, the multiple beta barrel, it will go like this. Now, if you look at, this will be the lipid bilayer and the barrel will stay inside this. So, if we have this kind of barrel structure open and there will be a lot of hydrophobic amino acid which is surrounded by this lipid bilayer and lipid chain is protecting those hydrophobic amino acids. Now, suddenly if you remove all of those detergent, all lipid, what will happen? All this barrel will be hydrophobic residues, this barrel will try to come closer with each other. So, there will be one barrel here, so somehow another barrel will be attached here, another barrel will be attached here and there will be some kind of aggregation of this barrel like protein. So, they cannot survive without detergent or lipid membrane. So, the aggregation, there will be a different, there will be many different reasons. One of the reason is that okay, a protein is happy in particular one environment. So, something like that, it is happy in presence of detergent, it is happy in presence of glycerol or high salt concentration. If you suddenly remove that, then protein will come and interact with each other and some kind of aggregation it will happen. So, exactly last class I was discussing about this one. So, that lipid protein aggregation. So, you can see that lipid protein aggregation in this particular region. There are some isolated particle you can see here, but there is a huge aggregation you can see. again you can see a huge aggregation here. And isolated protein molecule, if you pay attention, you can see all these isolated protein molecules here, here, here. But there are some kind of clusters, small lipid protein clusters are also there, but majority are that aggregation. So if you pay attention to this particular sample, here again you can see some isolated particle where you can see the central there is a pore. So something, a pore is also there, you can see this kind of, but overall from this background it is very difficult to isolate them from here. So this kind of lipid protein aggregation, definitely it is a problem for sample preparation. It is not about the staining, because staining wise it is perfect, your aggregation part is very much clear, as well as on top of that aggregation you can isolate some kind of protein molecules. So as a staining, if you just consider stain, here it is good, but sample is bad because it is a lipid protein aggregation. So that we have to consider and we have to work on that particular portion. Now come to the protein aggregation. This is another problem when what we are discussing, that okay, so here you can see there are some places, some molecules there is no definite shape. So you can see many are there clustering. So here again we are expecting this kind of circular protein molecules if we can isolate them, but here concentration is high and proteins are most of the time they are coming together and clustering this kind of thing. So this is basically protein aggregation. So that I already discussed that why the protein aggregation is happening, because if there is a intrinsically disordered region, or if there are some portion it is surrounded by detergent micelle or glycerol or high salt is protecting that protein, and suddenly we remove all of this condition, like we remove the salt concentration, we remove the glycerol completely, and we remove detergent. So then protein will try to come and form some kind of cluster and then they will aggregate. So here those are the protein aggregation, you can see it here. So this is again it is a bad sample. If I just consider that okay, this is definitely a bad sample, so not good for any other purposes. But from this experiment what we can tell, at least from this experiment we can tell that so in this particular condition is not suitable for any other study. There is a high chance the protein will come together and form some kind of cluster. So far we are constantly showing you some kind of bad images, right? So starting from very light stain, then very thick stain, there is some problem, the sample is very heterogeneous. So lipid protein, when we talk about the antigen antibody interaction, I mentioned that overall staining is very good. Even there during the lipid protein interaction also I mentioned that okay, overall staining is good, but the sample is bad because it is either some kind of aggregation or antigen antibody reaction. We can see some complexes, but then lot of free antigen, lot of free antibodies are there. So that is a problem again. So now what is a good image? Let's take a look couple of good images and then we will discuss about the application of this negative staining technique. So one of the good image I can say that okay, this is one of the good image where you can see very nice background. So background is very clean. So and the protein white, as I mentioned multiple times that white is a protein molecules for the negative stain, and from these images you can easily distinguish our protein sample here. They are very much isolated. There are some cases there are 2-3 proteins molecules will come together and cluster. So that is one problem you will always remember that. So 5 to 10% this kind of aggregated molecule you will get it. So you do not have to worry about that kind of small aggregation. But if you look at the image, if you can see your protein molecules very clear, the background, and you can see their morphology properly, that means that is a good data or good negative stain. So now come back to this slide and look at that. What I was talking about here, you can see there is some kind of elongated molecule. If you just consider this shape with this one, there is no match. So here 2 or 3 protein molecules are clustering together, so we can ignore that. Here also another one small part where 2-3 protein molecules are clustered. Here 2 molecules may be clustered, but most of the cases, if I just use a different color, it will be clear to you. So if I just use something this yellow color here, the black background, you can see that okay, those are my isolated. And if you look at this shape, they are almost similar, they are not looking completely different from each other. So those shapes are so close to each other, we can average them together. During the application time, we will talk about the averaging. So here you can see those are mostly isolated and good particle. So this is called one of the good TAME images, that where you have a very nice distribution of the stain, as well as you can isolate your protein molecules properly. So you can use this for further analysis, what I was talking about, the 2D classification or 2D classification. So similar looking protein molecules, will cluster them together and then we will calculate an average of that. When we will calculate the average from that, that is called the 2D averages, and that technique it is called 2D classification. So when we have a very nicely distributed protein molecules, so that will really help us to pick the single molecule and then make a averages of those protein molecules. Now take a look at another sample. So now in this particular sample, you can see this is basically a spike protein from SARS-CoV-2 coronaviruses. This is the SARS-CoV-2 coronaviruses. This is the spike protein. Spike protein generally it will be a trimeric structure, and the spike protein is attached with some antibody. So spike protein is antigen here and some antibody we incubated. So overall spike protein structure, many of you are familiar with this spike protein structure. So if we just consider that how it should look like, generally it has one protomer like that, then another protomer and then trimer. So from top it will look like a triangular shape, and otherwise it will come one protomer and another protomer. So this is a trimeric shape, you can see it like a flower. So here if we look at this particular image, this spike protein is incubated with an antigen, with an antibody, and this antibody is attached here. So in this entire image, if you look at this particular image, this entire image, it is very much isolated. You can see the background and very homogeneous sample all over. You can see there is not too much heterogeneity here. So this is called one of the good negative stain image, where you can isolate the sample and protein clearly, and you can see those molecules, and those molecules you can use it for 2D classification. That 2D classification we are going to discuss in the next classes. So here you can see this cone shape, lots of molecules are there all over the image. So whenever you will try to get a good image, try to get an image like that where these particles are well separated, well distributed, staining is nice, and you can see morphology of each and individual protein molecules here, and that is really helpful to do the further analysis. If the protein molecules are not separated properly, it will be very difficult to do any further study. Now, if you look at here, this is the another image what I am showing you here. This is a protein sample from Mycobacterium tuberculosis, and this protein sample, it has a tetrameric assembly. If you look at that, you can see similar kind of architecture. Many of this has some kind of rectangular shape. So, something like this kind of rectangular shape. If you just consider this is a rectangle. So, there are many samples, you can see they are all this rectangular shape, you can see it here. So, when you have this kind of images, you can tell that okay, I have a good sample, it is a very homogeneous sample, and also staining is so nice that you can see individual protein and the details of this individual protein, and the entire morphology or architecture is very much clear from this negative staining. So, this is basically one of the good image we have, and that we can consider for further study. And this kind of sample, if we have a proper concentration, and that we can take it for cryo-EM study for structure determination. Now, in this particular condition, I want to add one more important part, that what we discussed here for the negative stain, what is most important part, that we have a staining solution, and the staining solution means it is a either heavy metal stain. So, that heavy metal stain will scatter the electron beam strongly, and then protein sample is mainly carbon, hydrogen, nitrogen, oxygen. This carbon, hydrogen, nitrogen, oxygen molecule, it can transmit the electron beam easily, and we can get a white molecules. So, in negative stain we will get always black background and white protein molecules, and that will be exactly opposite when you will do cryo-electron microscopy. So, in cryo-electron microscopy we are not using any staining solution. It is completely normal condition where our protein is embedded in our buffer solution. And if you just consider our buffer solution and the concentration of the protein, or the size of protein, or the overall element of the protein. So, protein has carbon, hydrogen, nitrogen, oxygen, sulfur, sometime metal ion, and rest are the buffer. So, definitely protein's density is much higher than our protein, our buffer solution. Then if we consider that electron scattering will be much higher in the case of protein, and that is the reason in cryo-electron microscopy, when you will discuss about that cryo-EM images, you can see that overall protein appears as black in a white background. So, that is one of the major difference. Other two major differences are there. So, one is that for negative staining we are using a staining solution, and in cryo-electron microscopy we are not using any staining solution. Most of the time it is in a physiological condition. The third one is that in the negative stain your sample is collapsed or flattened. Why it is collapsed or flattened? Because the technique itself, we are using we are preparing our grids and then keeping it at dry condition or desiccator, and when the sample is completely dried, we are taking this sample and insert inside the microscope, and inside the microscope it is under high vacuum, and most of the sample will be in a dry condition. So, definitely the sample will be collapsed. But in cryo-electron microscopy we will prepare the sample in such a way that our protein will stay inside in a liquid condition. So, it will be embedded in amorphous ice. So, it will intact its overall morphology. So, that is one of the major difference between the negative stain and the cryo techniques. But what is the advantage of this one, the negative staining, you need only that negative staining solution and you don't need very high end equipment to prepare that sample. You just need an electron microscope, and then rest of the sample preparation techniques is much easier and very straightforward. You don't need very high concentration sample. Generally 0.1 mg per ml sample concentration is good enough, and 10 microliter sample, 10 microliter volume of sample is good enough for negative staining. Where for the cryo-electron microscopy you need a very high concentration, at least 1 to 2 mg sample is required, and volume is also higher, volume is required, and also you need a specific equipment to prepare the cryo sample, which is not required for the negative staining. That is the major difference. Now we discuss about the bad staining, good staining. Now how do we implement these methods or techniques in our research, or what kind of information we can extract from this negative staining technique? I already discussed about how from this small experiment, that aggregation, not aggregated sample, or bad imaging, high concentration. So from there we can get some information that we can use it for further study or optimization purposes, but actual application in next class we will discuss about that. And next class we will discuss about the application of the negative staining, and thank you all for your attention.